Although this, what's depicted here is a mutation that occurred randomly in a person who has skin cancer. The event reveals an organization of DNA where we have separated and rejoined the DNA from two genes and show that genes are composed of two parts. The transcription unit, the business part of the gene that can make the PDGF protein, and a regulator, a promoter that can regulate where, when, and how much of the gene gets made. And so what we've done is we've used the skin collagen gene promoter to accidentally stimulate synthesis of the PDGF gene in the skin cells, like that. This woman probably has one problem, and either she may have inherited that from mom or dad. And let's say she inherited it from mom. That's an idea where you'll often hear that families have a certain type of cancer that runs in their family. So this family could have a certain type of skin cancer that runs in their family, and it's because they have a mutation that makes them produce PDGF in skin cells and cause uncontrolled cell growth. And so that's the idea of inheriting it. Alternatively, maybe this woman got exposed to some X-ray, some ultraviolet rays at the beach, and she has a brand new mutation, damage caused by UV light rays, like that. Okay, so in either case, it was an accident that occurred, but it reveals this feature of DNA, and so I made this just little cute drawing to show this idea that the DNA of one gene can be like a hand, and we can split it apart. Genes are composed of two parts, the transcription unit that's going to be dealt with in chapter 17 and the promoter that'll be dealt with in chapter 18. And here's a figure from chapter 17, shows a nice summary, and really here's the pathway of gene expression. So we've got the information in the DNA, it gets copied into RNA, and RNA that gets used by the ribosomes is called messenger RNA, one type of RNA. There are other types of RNA that get made from the DNA. Messenger RNA or mRNA in the cytoplasm binds to a ribosome, and chapter 17 tells the story of translation. Chapter 18 tells the story of transcription, which occurs inside the nucleus, and transcription is about regulation of gene expression, switching a gene on or off. So we got those two stories, making insulin and regulating the production of insulin. And so I posted on our website a handout, and the handout is about molecular genetics, and it's about DNA as a molecule. So we say the DNA has genes, and this stretch of the DNA is the gene, and the gene is composed of two parts. So DNA, genes, and the promoter and transcription unit are very physical. So that's one reason that mutation could cause skin cancer is a good example because it's very physical. We can see how the DNA got torn apart and joined back together, just like pages in a book can get torn and joined back together. So here's just some facts from this handout. DNA is physical, it's linear, like words in a book, organized into clusters of information, and like a word. A difference between a gene and a word is that genes are very long, they are tens of thousands of letters long, of A, G, C, or T patterns, and those letters are called nucleotides. So the monomer building blocks of DNA, back to chapter 5, are these macromolecules are made up of nucleotides. This is DNA. RNA uses the letters A, G, C, and U. So the difference between DNA and RNA. And gene is like a word. It's kind of an organizing pattern in a book of genetic information. It's long, a lot of information. So this plan is called the genotype, and you inherited a genotype from your mom and your dad. Then the phenotype is the product of that plan. So you inherited a plan from your mom and dad when you were a fertilized egg. The plan was there, and then it got played out over time to produce a human, you, and you are the phenotype. So a couple of terms used in genetics that we'll talk about a lot. And a key concept is that you got, like we said, in a diploid cell, of which most of yours are, you've got one copy of mom's DNA and one copy of dad's DNA. And they stay separate. They're separate, but in the phenotype, they tend to blend together to get a blended outcome. And however, then if you have babies, you'll send either mom's copy or dad's copy onto your child. So we have this genotype being separate and staying separate over generations, whereas the phenotype is a kind of blending. And we can deal with that right now in this example. Here's aquaporin. Back to aquaporin, we can pick any protein. And I mentioned before, but it's worth mentioning again, that in the book and in a lecture, when we talk about aquaporin, any protein, we just draw one of it. But on a given cell, there are thousands of aquaporin. You need a lot of it to get the job done. And the next thing is, where's that coming from? Well, there's some RNA that's being used by a ribosome to make protein and make these. Now, that RNA comes from the DNA, and in the nucleus, we have DNA from mom making some of these. We also have DNA from dad. And that DNA from dad also makes RNA. It's grabbed by ribosomes to make proteins. And so if you look how aquaporin is doing its job of letting water go in and out of the cell through osmosis, this overall effectiveness of aquaporin is a kind of blending of mom and dad. It's a kind of averaging, you know. What if mom's aquaporin was great and dad's was no good? So it'd be an average between basically 100% and 0%. And then you'd say, okay, so on average, this cell's working at about 50% of aquaporin function. And is that good or bad? How bad is it? Like that. And it turns out we have to talk about genes on a case-by-case basis. There's no way of knowing that if a person had 50% of the normal aquaporin, if they'd be sick or have any kind of symptoms like that from this blending to produce the phenotype. Okay, so we'll talk a lot about that. Here's something from the handout just to show that the DNA code, a chromosome looks like this, and scientists can now read this code through a method called DNA sequencing. And billions of DNA letters are in databases available to look, just like you do a word search in a Word document. You can do a search to look for certain patterns of letters. And there are many of those sequences available on the web. So you could, for example, look for the insulin gene in pigs by looking at the pig genome and searching through it. All right? So we're not used to looking at these kind of letters. We're used to look at these kind of letters. It's the same thing. This is a language. This is a language. Then there are no spaces in the DNA language in our genes. And so I've underlined here what that would look like. So imagine that a book doesn't have any spaces and the letters just are continuous, but only some of the letters are spelling out a word. Some of the letters are organized into genes. And the genes have two parts. One part is the business part of it. That would be equivalent to the transcription unit. that gets the job done. It's the actual word. The transcription is the actual sequence that is used by the ribosome to make insulin. So we've got the word, and then here's the promoter. Somewhere else, there's probably another one. And so each gene is composed of two parts. And the story plays out when both are used. So you go to the first word, the quick brown fox. And that's how the promoter works. It regulates where, when, and how much of the transcription gets made. And so it's through the promoter that tells the story, that plays out the story of the gene's function. And so when we read it out in proper order, we get a story like that. So that's how it's organized. And at first, the DNA and genes are very confusing, but this is the bottom line of what's happening. All right. So we've got more to say about the homework, but I think we'll take a break at this point in this mini lecture. We just define this two parts of every gene. Good.